So even this during translation of proteins, the first two codons of the mRNA enter the ribosome and tRNA molecules, that is with amino acid attached, that have complementary anticodons to the first two codons of the mRNA bind to those codons. A peptide bond forms between amino acid carried by two tRNA molecules, and dipeptide is transferred to the tRNA in the A site. Consider the sites on the ribosome, the three sites. The ribosome moves along the mRNA by one codon, bringing the third codon into the ribosome. Consider that the protein making process is on the way. At the same time, the free tRNA exits the ribosome from the E site or exit site, and tRNA with the dipeptide moves into the P site, peptidyl site. A tRNA with complementary anticodon binds with the third codon and brings its amino acid into position next to the second. A peptide bond is formed between second and third amino acid, the same process. The whole process is repeated until a stop codon is in the position and translation is stopped. When the translation will terminate or stop is when the stop codon encountered one of the three stop codons. TGA, TAA, and TAG are the stop codons we have discussed. Note that the translation of the mRNA code into a protein molecule requires energy. Okay? Energy in the cells are in the form of ATP. That energy comes from the hydrolysis of GTP or ATP. This is not from the GTP. Not from the ATP. It is from GTP. In this process, the energy source is GTP, not ATP. So this is the ribosome, then large subunit, and there are the two subunits. A small subunit is here. Then generally P site and A site is considered in this process. The protein synthesis is going to take place. Then mRNA codon joins the ribosome. Okay, mRNA joins the ribosome. The first codon, the first triplet here is AUG. Then the first tRNA comes, then initiation. The first process of protein making is initiation, starting the protein making process. Then having anticodon, UAC, the complementary anticodon for the codon AUG is UAC. Then it attaches to the ribosome. Then hydrogen bonds between the bases forms. Then the next amino acid will be brought by this transfer RNA. Then the second transfer RNA with amino acid 2. Then the bond formed between the amino acid 1 and amino acid 2. Peptide bond will be formed. Then the third amino acid will come. Then the first transfer RNA leaves the site. Okay, at the exit site, it is exiting the ribosome. Then the bond between the second amino acid and the third amino acid formed. Then the fourth amino acid is coming. Then the second tRNA is leaving the ribosome. Then the process will continue in such fashion until it will finish the codon on the mRNA. So here you can see about after it stops here amino acid AA for amino acid 199. So you can consider that how many amino acids are here? 198 amino acids. Okay, for one protein. Then finally termination is here, or ending, because here is UAG, the stop codon is here. There is no amino acid for such codon. There is no anticodon for this stop codon, so there will be termination. Then the product is the protein. Though it is similar to eukaryotes, there are some differences, like prokaryotic cells have no nucleus. Prokaryotic mRNA does not need post-transcriptional process because it only contains exons or coding genes, not on or non-coding genes. That is the difference between mRNA in eukaryotes and mRNA in prokaryotes. The prokaryotic genetic makeup is just pure coding sequence of genes, that is exons. There is no post-transcriptional process. There is no modification of the transcripts. After the mRNA is made, there will be one modification. It will not be used as a raw. It will be modified. There is no such process in prokaryotes, but it is there in eukaryotes. Because the eukaryotic genetic makeup is very complex, having introns or non-coding genes and other non-coding factors. Transcription and translation are coupled, they go together. mRNA translated at one end of ribosome while it starts transcribing from DNA at other end. They are coupled. What is transamination? This is a process of amino group of an amino acid is removed. Okay, a removal of the amino group from the amino acid we call transamination. Then it will be transferred to a keto acid, then it becomes a different amino acid. That is keto acid. For example, pyruvic acid, that is keto acid, will be converted into alanine, which is amino acid by transamination. Note that not all amino acids are produced by transamination. Only those we obtain from our food can do transamination. They are called essential amino acids. Okay? The amino acid which will not be produced within our body and we have to take through our food are called essential amino acids. That is it. Control of gene expression. The chemical processes or the reactions within body in living organisms are controlled process or regulated processes. The same is true in the gene expression. When you say gene expression, simply conversion of the genetic code in DNA will be converted into the protein that we call gene expression. This process is controlled or a regulated process. Genes are switched on, switching on. You switch on your light in your room. Like that, genes are switched on by what? Transcription factors. These are proteins. Which are proteins that bind to a regulatory sequence of DNA near to the gene they influence. Okay, where they found this around the gene, they do their job. They operate in the following way. That is, the transcription factors that are proteins bind to a promoter sequence of DNA near the gene to be activated. We call such site promoter region or promoter sequence. Then RNA polymerase binds to the DNA. Okay, the enzyme, the DNA dependent enzyme, that is RNA polymerase, binds to the DNA. Then it makes what? Transcription factor complex. Okay. The RNA polymerase activated and moves away from the DNA along the gene. The RNA polymerase transcribes the antisense strand of the DNA and the gene is now being expressed. Like transcription factors that promote gene expression, there are factors to repress. Repressing means preventing or then switched off gene action. Short interfering RNA, SI for short interfering are one example of repressing factor. These are usually very short, only 21 to 23 nucleotides. That means A, T, C, G, A, T. You can count any kind of such nucleotides, 21 up to 23, very short, double-stranded. They do not act on the gene itself, but interfere or silence the mRNA once it has been transcribed from DNA. Their action is not on the gene, okay, after the transcription is made. Is post-transcriptional interference, because after transcription process is made, we call this is post-transcription. So this process is after transcription is processed. If mRNA is prevented from translating its codons into amino acid, then the protein for which the gene codes for cannot be built, then silenced. That is simple statement you can easily understand. If there is no translating, then there is no proteins. Mutation is the alteration of the nucleotide sequence of the genome of an organism, virus, or extrachromosomal DNA. It is simply a change. Alteration means change. So changing the nucleotide sequence. Nucleotide sequence means adenine, cytosine, guanine, cytosine, guanine, cytosine, cytosine, thymine. That is the sequence of what? Nucleotide. So the change in that is called mutation. Mutation could occur spontaneously, that is accidentally during duplication or DNA replication. Or there may be other factors for mutations. There are a lot of factors. One factor is this one. Mutations are rare events, okay? Rare, they are not common. Rate of mutations can be increased by carcinogenic, that is carcinogenic chemical in tobacco smoke. Okay, smoking tobacco is bad, don't smoke, because it will cause mutation, then it will cause what? Cancer. Carcinogenic means cancer causing. High energy radiation, that is UV radiation, X-ray, these are also the factors which can increase mutations. There are many different ways that DNA can be changed, resulting in different types of mutation as described below. There are generally two kinds of mutation, gene mutation and chromosomal mutation. We call gene mutation as point mutations. This is changes that involves a single base, that is why it is called point. There are several types of point mutations. Some of these are substitution, addition, and deletion. Substitution means, from its name, one base will be substituted by another or replaced by another. Addition means an extra base is added. Deletion means a base is missed out during DNA replication. Chromosomal mutations, situation where part or segment of a chromosome sequence of DNA is disturbed, or a chromosome is missed or added. Types of chromosomal mutations, there are different kinds of chromosomal mutations. Which one is intense or which one has a large effect is the chromosomal mutation, because it will affect a lot of sequences of nucleotides compared with the point mutation. There are the kind of chromosomal mutation. One is euploidy. Eu means true, having a chromosome number that is an exact multiple of the haploid number. One example in Euploidy is Polyploidy. Poly means many. That is the condition in which a normally diploid means di means two. Ploidy refers to the chromosome. So diploid having two sets of chromosome. This diploid cell or organism acquires one or more additional sets of chromosomes. For example, triploid, tetraploid. For example, N is, let's say, 23 in human, N is 23. 2N is 2 times 23, which is 46 chromosomes. 3N means 3 times 23, 69, that is triploid. 4N, 4 times 23, which is equal to 92, that is tetraploid. These are, we call euploid in the set. The increment is in the set of haploid member. Aneuploidy, which is the opposite of euploid. This is a condition where an organism or a cell lost from or added to one or more chromosomes to the normal set of chromosomes. For example, 2n plus 1 trisomy in the human 46 plus 1 is 47. In such condition, there will be Down syndrome. This is a kind of defect, or we call Down syndrome. This is named by the scientist Dr. Down. 2n minus 1 we call monosomics. The kind of defect is Turner syndrome. The chromosome will be 46 minus 1, 45. 2n plus 1 plus 1, double trisomy. Trisomy is adding one chromosome. If there will be adding two chromosomes, we call double trisomy. 2n minus 2, nellisomic, organism that lost one homologous chromosome. Giddu syndrome, this is mental retardation caused by part of deletion of chromosome number 5 of man. Okay, there are how many chromosomes? 46. 23 pairs or 46 chromosomes. If there will be deletion of the fifth chromosome, we call Giddu syndrome. Giddu syndrome will cause that kind of disease, that is mental retardation. Consequence of gene mutations. Mutation on body cell, that is non-sex cell, cannot be inherited, but may result in one of the following. It may be harmless, there is no effect. It may damage the cell. It may kill the cell, or it may make cell cancerous, which might kill the person. Mutations in different genes will obviously produce different effects, but two types of gene are important in regulating cell division, okay, and preventing the formation of tumor. The first gene is Proto-oncogenes. The name is gene oncogenes. Onco for cancer. A gene involved in normal cell division or cell growth, that is proto-oncogene, is a kind of gene involved in normal cell growth. When these genes are mutated, they often become active oncogenes, which stimulate cell to divide in an uncontrolled manner. The cell will divide many times, or so it will cause what? Cancer. Tumor suppressor genes. The other second gene is the tumor suppressor gene. Recognize this uncontrolled cell division and act to suppress cell division. Okay, it will recognize the mutation of the first gene, then it will suppress cell division, prevent cell division. If these genes mutate and become inactive, a tumor will form as uncontrolled cell division continues. Tumor, what is tumor? It is a mass of cells created when cell replication gets out of control and cause cancer. Thanks for watching.